What if I told you there's this secret clock ticking away inside your head? And no, I'm not talking about one that tells you when it's lunchtime. I mean one that makes, well, understanding the world itself possible. We're about to pull back the curtain on this incredibly critical, but honestly often overlooked part of our brains.
Okay, so let me ask you this. What if you could hear a sound, but your brain just couldn't make sense of it? Like, picture this. You're sitting with a friend, they're talking, and you can hear their voice just fine. The volume's perfect. But the words, they're just a scramble, a jumble of noise. See, this isn't a problem with your ears. Nope, it's something way deeper. It's a timing issue. Something's getting lost on the way to your brain.
So this really puzzling condition, it actually has a name, auditory neuropathy. And it's fascinating because the ear, it does its job perfectly. It detects the sound waves, no problem. But then the information gets corrupted somewhere in delivery. The absolute core of the problem is this loss of what scientists call temporal acuity, which in plain English just means the brain's timing is completely off.
All right, so to really get why this timing fails, we've got to look at the wiring itself. You can think of it like this massive biological superhighway, just buzzing with high-speed signals. Let's dive in and check out that neurowiring.
So your brain, it's just this incredible network of billions and billions of neurons. And they're all talking to each other, right? They do it by firing these tiny little electrical messages back and forth. Scientists call them action potentials. And this isn't just for fun. This is literally how you think, how you feel, how you perceive, well, everything. Now, for something super complex like understanding speech, these signals have to be lightning fast and perfectly coordinated.
So how in the world does the brain pull off this incredible speed and precision? Well, it has a secret weapon, and it's called myelin. The best way to think about it is like the plastic insulation you see on an electrical cord. This fatty stuff literally wraps around the wires of your neurons, their axons, and it stops the electrical signal from just fizzling out. And because of that, the signal can travel way, way faster.
And wow, this image just perfectly shows what we're talking about. Take a look. In a healthy auditory nerve, you can see the axons. Those are the neural wires, and they are just completely coated in that critical myelin, which you see here in green. That dense wrapping, that is the secret sauce. It's the key to getting a fast, reliable, and perfectly timed signal.
Okay, so we know it's important. But how do we know for sure just how important this insulation is? Well, scientists found the perfect way to figure it out, thanks to this really fascinating natural experiment. It's kind of a tale of two very, very different rats.
All right, so on one side of our experiment, we have our control group. This is a normal, healthy Long Evans rat. It's got a perfectly standard, myelin-coated nervous system. But then, on the other side, we have our variable. And this is where it gets interesting. This is the Long Evans shaker rat. It's a special mutant that, just because of a genetic quirk, is born without that compact myelin insulation in its central nervous system.
And here it is, the visual proof. It's just so clear. On the left, you've got the healthy auditory axons from the normal rat, just dense with that green myelin. But then you look to the right at the mutant rat, and it's just gone. The wires are there, but that critical insulation is completely missing. I mean, this sets up the absolute perfect test, right? What happens to the signal now?
So the big question is, what did the scientists find when they compared them? Well, the results were pretty staggering. They revealed just how slow, jittery, and unreliable these signals become when you take away that insulation.
So let's get right to it. What happens to the brain's high-speed messages without this crucial insulation? I mean, what really happens to the quality of that signal? Does it get slow, jittery, unreliable?
The first piece of evidence was huge. The signals were 3 times slower. That's right. The speed of that action potential, that electrical message, was 3 times slower in the rats without myelin. That's just a massive drop in speed. It's like turning a superhighway into a bumpy country road.
But it wasn't just about speed. The precision of the signal, it completely fell apart. Scientists have a great word for this, jitter. And you can think of it like a drummer who just can't keep a steady beat. Boom, 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 boom. It's all over the place. And as you can see from this chart, the mutant rat had about 4 times more jitter than the normal rat. So the signals weren't just slow, they were arriving at completely inconsistent, unpredictable times. Okay, so we've got slow signals, we've got jittery signals, but here's the final test. Can these signals even keep up under pressure? The researchers basically decided to give these neurons a stress test. They pushed them to their limits with high-frequency stimulation to see what would happen. kind of mimicking the intense demands of processing something like speech. So first up, they hit him with 100 Hz stimulation. That's a pretty fast pace. And at that speed, both the normal and the unmyelinated neurons, they fired just fine, no problem. But then, they cranked it up to 300 Hz. And that's where things fell apart. At this very fast pace, the unmyelinated neurons just started to fail. They couldn't keep up. The signal would just drop out. So now we know they're slow, they're imprecise, and they're totally unreliable under pressure.
Okay, so that's all super fascinating science, right? A tale of two rats. But why does myelin matter? What does this mean for us? Well, this is where we connect it all back to the human brain and why this is so incredibly important.
Here's the thing. For our brains to decode something as complex as human speech, those signals need to arrive with almost perfect sub-millisecond precision. But, just like we saw in the rats, when that signal gets slow and jittery and unreliable, the message just gets garbled. It turns into noise. And that helps explain exactly what's going on with auditory neuropathy. And it doesn't stop there. A similar kind of signal disruption is a huge factor in diseases like multiple sclerosis, or MS.
You know, the researchers who did this study, they put it perfectly. They said proper myelination is essential for precise presynaptic AP firing that minimizes both spike jitter and failures. So it's not just some nice-to-have bonus feature. It is absolutely essential for keeping those brain signals fast, precise, and reliable. It's what stops the jitter, and it's what stops the signal from failing completely.
It really makes you wonder, doesn't it? If this level of perfect timing is so critical for just hearing, what other incredible abilities of our brain depend on this microscopic insulation? Think about it. Complex thought, coordinated movement, maybe even consciousness itself. How much of who we are is built on this tiny, fatty layer wrapped around our neurons?